Hello, everyone. As Maya mentioned, I'll be presenting the work I've done this summer at the Spraggins Lab regarding multi-omic characterization of the human pancreas. So very quickly, a brief introduction on the pancreas. It's a unique organ situated between the spleen and the small intestine, and it has two main functions. One is to produce the enzymes and other secretions needed for digestion, and the other is a hormonal function where it produces and regulates the release of hormones that affect blood sugar and appetite. The exocrine aspect of the tissue comprises of about 95% of it, and it consists of acinar cells, which are responsible for producing those digestive enzymes I mentioned, duct cells, which are responsible for carrying those secretions to the small intestine, as well as connective tissue and vasculature that is found in all organs. And the endocrine or hormonal aspect of the pancreas is made up of islets, which are small regions of localized cells that are randomly distributed across the pancreas. And each cell in that islet is specific to the production of one hormone. Most notably, alpha cells and beta cells produce glucagon and insulin. And on the right here, I have a figure showing the imaging techniques that the Spraggins Lab uses in collaboration with HuBMAP in order to better understand the pancreas. So the Spraggins Lab uses imaging mass spectrometry along with other multimodal imaging technologies in order to gain a better understanding of the tissue microenvironment, both in a bulk and a spatial context. And here I just have a general workflow that someone might do, which consists of autofluorescence microscopy, imaging mass spectrometry, immunohistochemistry, and stain microscopy. And this is an image of a human pancreas. So this summer, I wanted to research how different fixation techniques might alter our imaging analysis capabilities in the context of the human pancreas. And to do that, I used a multimodal workflow to compare data generated from fresh frozen tissue and paraformaldehyde fixed tissue from the same pancreas donor. So fixation is a way to prevent tissue autolysis and the degradation that naturally occurs due to enzymatic reactions and other metabolic processes after death. Paraformaldehyde fixation is a way to stop these reactions from occurring by cross-linking amino acids and lipids. So the pros of fixation are that you get a better preserved tissue and intact cells and molecular structure. However, because you are modifying analytes after death, there may be some identification or signal loss of your target analytes depending on what methods you're using. And in general, it's worth noting that the Spraggins Lab sticks with fresh frozen tissue because of the amount of imaging technologies that we use. It's best to use an unaltered tissue that's compatible with with all technologies. However, with the pancreas, this research question is worth looking into due to its unique nature, since it produces the digestive enzymes needed for the entire body. It tends to digest itself and rapidly degrades after death. Here I have an antibody stain of an islet as staining for beta cells and alpha cells. As you can see here in the PFA fixed tissue, our islet is much more intact and localized and looks like an islet compared to our fresh frozen, which looks very degraded and very delocalized. So to research this question, I used three methods, one being matrix-assisted laser desorption ionization imaging mass spectrometry, or MALDI-IMS, and the other two being liquid chromatography coupled to mass spectrometry, one for lipidomics and one for proteomics. And I have this figure here to show how the three methods inform one another. So if our goal is pancreatic cell analysis and mapping, then we can use MALDI-IMS to view our analytes in a spatial manner. And then we use LCMS for lipidomics in order to inform us on our identifications, so we know what we're looking at in our MALDI images. And then LCMS proteomics exists as a supplementary mode to compare another analyte, which would be proteins. Very briefly, LCMS consists of a liquid chromatography instrument, which uses stationary non-polar phase and mobile polar phase to separate analytes in a sample based off of their hydrophobicity. More hydrophilic molecules are eluted faster into a mass spectrometer, which further separates these molecules based off of weight. Multi-IMS is a type of mass spectrometry that consists of applying an organic matrix to a mounted tissue section, and upon insertion into the mass spectrometer, a laser is fired at that tissue in a raster pattern, and this causes the matrix and the sample underneath to ionize in a plume, and then analytes are separated based off of weight and then detected. And later on, a computer program can then generate heat-like maps of each ion so you can view the distribution across the tissue. So moving on to our results, our proteomics data revealed that more unique peptides were detected in our fresh frozen tissue than our PFA fixed tissue, as seen in this Venn diagram. However, regarding the proteins that were detected, on average, PFA tended to have better peptide coverage for it. So this is in line with what we think of when we think of fixation. While you may be getting some identification loss, the proteins that you do identify will be better preserved in the tissue. Our lipidomics results show very similar metabolite detection as we see in these Venn diagrams. It's almost 100% similar. And the difference moving from lipid identification to lipid is in intensity. So I have 4 lipids here and they're box plots. And as you can see, the intensity varies moving from lipid to lipid. Our MALDI IMS results are here. I have the pre and post IMS autofluorescence images just to show how we use autofluorescence to inform and help us choose the region of the tissue to image. We try to choose a very diverse area as seen in the pre-IMS images. We could see exocrine tissue throughout the sample as well as vasculature outlined in blue, and then areas where there's bright spots are indicative of islets. And then K-mean segmentation is a tool used to cluster your mass spectra in a localized manner. And I just have the results from fresh frozen and PFA fixed just to show that K-mean segmentation can be used as an unbiased tool to express tissue heterogeneity. And here I have a zoomed in area from our post-IMS autofluorescence just to show that we are collecting images at 10 micron resolution as seen from the laser marks. Here, I have a couple of side-by-side ion images so we could see the different distributions. Fresh frozen will be on the left and PFA will be on the right. And as you can see, there is some difference regarding intensity and localization. In particular with these ions, we can see that in the PFA fixed, the ion intensity tends to be more localized or concentrated compared to our PFA fixed, which looks a little bit more diffuse. So in conclusion, the data that we collected thus far shows that there is a difference regarding protein identification when comparing fresh frozen and PFA fixed tissue. However, with our lipidomics workflow, there was little difference in our lipids identified, and the differences were in intensity and potentially distribution. Some future directions that we could do would be to replicate this experiment with the same donor and perhaps a different donor just to validate our results. We would also benefit from incorporating antibody and H&E staining into our workflow in order to further inform our MALDI IMS and autofluorescence images, just so we know better structures that we're looking at. We could also do more research into the reagents and materials that are introduced in our fixation process to make sure that we aren't introducing any contaminants into our data. And lastly, we could replicate this workflow in the head, body, and tail of the pancreas, which are 3 different regions, and compare bulk and spatial analyte profiles to see if there's any differences. And in this slide, I just have a compilation of all the data that we collected during the summer, just to show that we have a lot to work with and more than what I could cover in this presentation. During this internship, I learned quite a lot. I learned everything that I did at this lab was pretty much new to me. I learned a lot regarding tissue handling and imaging, as well as data analysis and presentation skills. And I cultivated really great mentor relationships, and just this helped me a lot regarding my academic career. I'd like to thank the NIH for funding this internship and our supervisors for being along with us every step of the way. I'd like to thank the Spraggins Lab for my awesome experience here, and in particular, I'd like to thank my mentors, Dr. Angela Cruz and Dr. Maddie Colley, for helping me out with this project. And lastly, I'd like to thank Dr. Strikalova and Rachel Leo Galvin from the University of Florida for providing great seminars throughout this internship. Thank you.